Histopathology, fresh tissue examination.
So, there are four main methods of fresh tissue examination, and these are the teasing or disassociation, squash preparation, smear preparation, which consists of four sub-methods, the streaking, spreading, pull apart, and touch preparation, and the last method would be the frozen section. But what is the best method for tissue examination? Well, the methods of tissue examination may vary according to the structural and chemical components of the cells to be studied, the nature and the amount of the tissues to be evaluated, and the need for an immediate examination of a tissue structure.
Examination may be done on fresh or preserved tissues depending upon necessity. Fresh tissues have the advantage of being examined in the living state, thereby allowing protoplasmic activities such as motion, mitosis, phagocytosis, and pinocytosis to be observed.
Now, let's head into the procedures.
Teasing or dissociation. Selected tissue specimen is immersed in isotonic salt solution in a petri dish or watch glass. The tissue is now carefully dissected with a needle and separated by direct or zigzag spread using an applicator stick. Selected pieces of the tissue are transferred carefully to a microscope slide and mounted as a wet preparation underneath a cover glass, care being taken to avoid forming bubbles. This can now be examined under phase contrast or bright field microscope. It permits the cells to be examined in the living state, however, preparations are not permanent. Squash preparation or crushing. Small pieces of tissue, not more than 1 millimeter in diameter, are placed in a microscopic slide and forcibly compressed with another slide or with a cover glass. If necessary, a supravital stain may be placed at the junction of the slide and the cover glass. Streaking. With an applicator stick or a loop, the material is rapidly and gently applied in a direct or zigzag line throughout the slide, attempting to obtain a relatively uniform distribution of the secretion. Remember that too thin or too thick smears must be avoided.
Moving on, we'll proceed with the smear preparation, but before placing the specimens, we have to make sure first that our slides are clean, dirt-free, and is grease-free. Let's continue, starting with the blood smear. Now, blood smear is a type of smear preparation where blood is spread lightly over a slide by spreader. Don't forget to label the slide with a lead pencil. Next one is spreading. A selected portion of sample is placed into the moderately thick film and is teased with an applicator stick. Don't forget to label the slide with a lead pencil. Next one is the pull apart. Pull apart involves placing a drop of specimen in one slide and is faced in another slide. Don't forget to label them. And lastly is the touch preparation, where you place a freshly cut piece of tissue to a clean slide and is pressed onto the surface, allowing cells to be transferred and be examined directly by phase contrast or be stained by light microscopy.
Paperboat making for histopathology. First, fold the index paper in half, vertically and horizontally. Then, fold the index paper by aligning the vertical and horizontal lines, forming a diagonal line. After that, fold the index paper and divide it into 3 rows. Then fold the index paper into 5 columns. Lastly, locate the acute angle and push or fold it accordingly. Do the same on the other side.
These are the major materials we use in manual fixation. First is the precision knife. Second is the scissors. And lastly, the forceps. So we have to cut the desired meat, place it in the gauze pad and wipe out the excess fluid. Observe the specimen microscopically and then measure the specimen, the width, the length, and the height. And get a sample about 2 by 2 centimeter. Once you are done cutting, arrange it by size. Place the tissue in the tissue cassette, but then gauze pad is used as substitute.
Hi everyone, today I'm gonna make a video for the bone decalcification. So now I'm gonna introduce to you the materials needed in this video. These are the materials needed in the bone decalcification. First is the bottle, empty bottle. Second is the vinegar. Vinegar is a mild acid soaking the bone. Its vinegar removes the calcium in which make it soft and bendable the bone. And lastly, the bone. This is a bone for chicken. In this video, we will observe for 3 days to make the bone soft and bendable. So now the procedure of this video is first put the vinegar inside the bottle. Second is the bone. So now we will keep this and observe it after 1 day, so we can check what will happen now the bone inside the vinegar. So now I've already replaced the bone inside the empty bottle together with the vinegar. And I will observe it after 1 day. So now I will observe it after 1 day. So now I will get the bone inside the bottle. As you can see, after day 1, the bone will bended little bit like this. So now I will replace it again inside the bottle, and I will observe it in the next day or day 2. So we can observe what will happen in the day 2. Today is day 3, and I will get the bone inside the empty, and I will see what will happen in the bone, if they are already bendable or not yet. We'll see it. The result of this bone is not yet already bendable. It's because we need a lot of time, days, to soak in the vinegar so that it will make a bendable bone. But the other one is already bendable little, not all, because we need a lot of time or days to soak in the vinegar. That's why it's not already bendable, the all bones. The other one is little bit also bendable. So bone decalcification is a softening of bones due to the removal of calcium ions, and can be performed as a histological technique to study bones and extract DNA. This process also occurs naturally during bone development and growth, and when uninhibited can cause diseases such as osteomalacia.
Protocols following histo techniques. 1. Labeling. 2. Fixation. 3. Decalcification or optional. 4. Dehydration. 5. Clearing. 6. Impregnation or infiltration. 7. Embedding. 8. Trimming. 9. Section cutting or microtomy. 10. Staining. 11. Mounting. What is tissue processing? Tissue processing is a procedure of removing water from cells and replacing it with a medium which solidifies, allowing thin sections to be cut on a microtome. Tissue processing is routinely done on an instrument called tissue processor. Tissue processing it can be subdivided into dehydration, clearing, impregnation or infiltration. What is manual tissue processing? In this process, the tissue is changed from one container of reagent to another by hand. Note: the processing, whether manually or mechanically, involves the same steps and reagents in same sequence. What is dehydration? Dehydration, or the removal of water. It is the process in which the water content in the tissue to be processed is completely removed by passing the tissue through increasing concentrations of dehydrating agents. Tissues are dehydrated by using increasing strength of alcohol. 70%, 90% and 100%. Water is replaced by diffusion. During dehydration, water in tissue has been replaced by alcohol. The next step, alcohol should be replaced by paraffin wax. As paraffin wax is not alcohol soluble, we replace alcohol with a substance in which wax is soluble. This step is called clearing. Materials: slide staining rack, dehydrating agent, specimens. Commonly used dehydrant: ethyl alcohol. The most commonly used alcohol used in tissue processing is ethanol. Various dehydrating agents: acetone, alcohols, butanol, shell dissolve, dimethoxypropane, DMP, and diethoxypropane, DEP, dioxane, ethanol, ethyl alcohol isopropanol, methanol, phenol, polyethylene glycols, tetrahydrofuran. Dehydration should be accomplished slowly. If the concentration gradient is excessive, diffusion currents across the cell membranes may increase the possibility of cell distortion. For this reason, specimens are processed through a graded series of reagents of increasing concentration. Excessive dehydration may cause the tissue to become hard, brittle, and shrunken. Incomplete dehydration will impair the penetration of the clearing reagents into the tissue, leaving the specimen soft and non-receptive to infiltration. How is the dehydration achieved? Dehydration is achieved by placing the tissue in graded alcohols to avoid damage to the tissues. For example, it starts with 70% alcohol, then 90 to 95%, and then 100% alcohol. The series of increasing concentrations is used to avoid excessive distortion of the tissue. A typical dehydration sequence for specimens not more than 4 mm thick would be transfer slide rack into the first staining dish containing 70% ethanol. Incubate for 15 minutes. Then transfer the slide rack into the second staining dish containing a 90% ethanol. Incubate for 15 minutes. Then transfer the slide rack into the third staining dish containing a 100% ethanol. Incubate for 15 minutes. Then transfer the slide rack into the fourth staining dish containing a 100% ethanol. Incubate for 15 minutes. Then transfer the slide rack into the fifth staining dish containing a 100% ethanol. Incubate for 30 minutes. Then transfer the slide rack into the sixth staining dish containing a 100% ethanol. Incubate for 45 minutes. And finally, transfer the slide rack into clearing agent dish containing xylene, then let it soak for 5 minutes with frequent agitation.